So insertions and deletions, because by definition we're either adding or getting rid of something, can involve more than just a single base. So even though they are an example of point mutations, because they can involve just a single base, we can also see more than just one base. So at the most extreme, they can include thousands of bases. So just whole sections of bases being added or deleted. And this can translate into whole chunks of chromosomes, which we'll learn more about in the next chapter. Note that when they involve a multiple of 3 bases, a multiple, so that could be 3 total bases, 6 bases, 9 bases, 12, they're not considered a frame shift mutation. That's because the series of amino acids we see isn't altered, but the types of them are. If, however, we see insertions or deletions that are not in a multiple of 3, so we see 1 or 2, 4, 5, any of those examples, we will see a frame shift mutation because the series in which those amino acids occur is completely different. So just keep that in mind, that some deletions and insertions that are non-multiples of 3 bases are also frame shift mutations. Some deletions and insertions, as long as they are multiples of 3s, they're still insertions and deletions. They still can have effect on phenotypes, but we don't call them frame shift mutations. So going back to table 12.4, we see that frame shift mutations alter the way that the code or information is read. Remember, they're caused by insertions or deletions of 1 to many bases, but it has to be a series that is not a multiple of 3. What happens when we do this, we still have the letters that, comprise the words that we saw, but because they're organized in a different way, the sentence or the final protein just doesn't make sense. So deletions are simply mutations that remove DNA. And those deletions can range from a single base to thousands of bases or huge parts of chromosomes. Many common inherited disorders actually are due to deletions. One example are just tiny deletions in the Y chromosome are enough to cause male infertility. So the Y chromosome is really small and isn't comprised of that many genes. So even small losses of information can have huge effects. Insertions simply add DNA. So we can have one base to many bases. And Gaucher disease is an example of a disorder that's due to an insertion of just a single base, because that insertion actually prevents the production of an enzyme that normally breaks down a component of lysosome in lysosomes, which ultimately ends up causing problems within the liver and spleen and neurological impairment. So pretty serious and devastating effects. There's another type of insertion called a tandem duplication, where you have a whole part of a gene that's repeated. And the whole part that's repeated usually ends up being right adjacent to, or at least nearby, the original sequence. So it's almost like you're typing and repeating the same word back to back. So there are 2 copies hanging out, and those have been associated with genetic disorders as well. Looking at our table 12.4, we see how deletions involve the removal of DNA. Remember, when it's just involving one or two bases or multiples thereof, we also can call them frame shift. But if they're multiples of 3, as we see in this example, we end up missing or losing an entire word that comprises a sentence. They can also occur on larger scales, right? We've already talked about whole chunks of information that can go missing. So even though we have parts of the original information, some of it is lacking or missing. Then we have insertions that involve the addition of DNA, and they're anything other than multiples of 3. They also cause frame shift mutations. But in this example, it's not going to cause a frame shift because we only have an insertion of 3 bases. So we add entire words. It's adding amino acids into a protein product that shouldn't be there. Interestingly, we can have different types of mutations that end up causing the very same single gene disorder. Here we have an example of familial hypercholesterolemia, so high levels of blood cholesterol. These mutations are altering the LDL receptor that's normally anchored in the plasma membrane. And the way that the receptors are altered just varies depending on the type of mutation that we see. So in our A example here, we have a very specific missense mutation. So there's a base substitution. We end up with a non-synonymous codon, meaning we end up with a totally new amino acid. So we have a missense mutation. And what this does is, even though all of the other amino acids are the same, because of different chemical interactions between or attractions between these component amino acid, it ends up bending or changing the shape of the receptor enough to impair its function. In example B, we have a nonsense mutation. So we have a shift, a non-synonymous codon that, rather than encoding a different amino acid in this case, is a stop codon. So we have a signal for a protein release factor. The protein is completely shortened because we have premature stopped translation. So here you can see we end up with a protein that's only 3 amino acids long. Clearly can't perform its function of removing cholesterol from the bloodstream. Finally, we have example C, where we have an insertion of 4 bases. So the 4 new bases that we see occur in a position higher up in our amino acid chain, but essentially it alters the reading frame. So everything from this point on is changed. And because the organization of things have changed, we have a different series of amino acids than we would have. We see different electrical charges associated with the amino acids that then alters the final protein form and ultimately its function. Any type of point mutation can really affect a gene's final product if it alters a site where the introns are normally removed during mRNA processing. So remember that during transcription, we're copying essentially all of the information into a transcript. But before it's done, we do some processing. And in the processing, we remove introns, so some of the information. Well, if any of these types of mutations, whether they be substitutions, insertions, or deletions, alter a site where we should normally remove an intron, they can affect the phenotype. So for example, the intron may now be translated instead of being removed. And an example of that is cystic fibrosis. It can also actually extend further within the segment and then be removed. So rather than being maintained, the intron is still removed from the transcript, but because it extends, it might take out a chunk of an exon that should have been included. And that ends up being referred to as exon skipping. So we may either add information that wasn't there before, or we may take out important information that influences protein production. There's a type of mutation that is called an expanding repeat. And this happens when a gene actually grows as a small part of a sequence is copied and added. So this is a series of repeats that keep being inserted into a DNA sequence. When this happens, the proteins become really long. And when proteins are elongated, they can actually cause cells to shut down. Interestingly, there are some genes within our genome that are especially likely to becoming expanding repeats. And those genes end up being repeated over time, and essentially the conditions associated with them start appearing earlier in an individual's lifetime for those individuals that inherit the mutations. If they have expanded repeats compared to their parents, they experience the onset of symptoms earlier in their life, and they may even suffer from increased severity of symptoms. And that's because we end up with increased length of the associated abnormal proteins. Back to table 12.4, this just shows how these expanding mutations kind of increase in length over time. So we can see that we're repeating a whole word, right? So we're starting by copying a piece of information. So we've got a small part of our sequence, in this case, just one codon that's being altered. And in the second generation, we've added the codon twice. And it doubles again for the next generation, where we have 4 copies of the codon. So in each successive generation, we are doubling the amount of codons that we end up seeing repeated. So those repeats are appearing in greater densities across generations of time. Myotonic dystrophy is an example of a genetic disorder that is due to an expanding repeat. And specifically, the repeat that we're dealing with is an expanding triplet. It's also called a trinucleotide. So a series of 3 bases. And this disorder is just one example of about 15 diseases that are all associated with expanding triplets. Interestingly, a repeat number of triplets that's less than 40 copies is actually stable. So it can be passed on in the next generation and you don't end up seeing any symptoms at all. But once you get more than 40 copies, those larger repeats become unstable. And they become unstable, growing with each generation. You end up seeing associated symptoms earlier and ones that are more severe. So these triplet diseases are due to the nature of the DNA sequence. So the bases of these repeated triplets end up bonding to one another in ways that normally the DNA wouldn't. So it alters the actual conformation of the DNA into different shapes, such as hairpins or loops. And when that happens, those loops interfere with DNA replication. DNA isn't able to replicate the way it normally would, and so those repeats continue expanding over generations of time. So they're just being added to successively. And when those repeats are actually translated, you end up with really long proteins, and those proteins end up harming the cells in different ways. So they can bind to different transcription factors, they can block proteasomes. So that means that when you have misfolded proteins, they're unable to persist. And they also can trigger early programmed cell death. So here we show a pedigree of myotonic dystrophy, an example of one of these triplet repeat diseases. And we see two things in this pedigree. So when you're looking at the age of onset, because this individual has fewer of the triplets repeated in the genetic code, the onset of the symptoms was older in adulthood, and the phenotypes were less severe, right? This is all because we're seeing fewer copies of the actual repeat. Well, these repeats keep accumulating and passing on to successive generations so that we see age of onset in the next generation, we'll say 20 or so years earlier, phenotypes that are more severe. These mutations get replicated and repeated more numerously in the next generation. So age of onset is much earlier in childhood and severe symptoms. In chapter 7, we read about copy number variants. So these are specific DNA sequences, so whole sections, that vary in number of copies from person to person. And these account for about a quarter of the genome. So typically they involve larger chunks of DNA that are inserted, deleted, or repeated. So not just a single base or a few bases like a single nucleotide polymorphism, but rather whole chunks of information are repeated. So imagine words that are being repeated in multiple places in differing numbers of copies. So these copy number variants may have absolutely no effect on phenotypes, and they actually contribute significantly to differences in among us. It just depends on how many we have and how many far away they are. They can, however, disrupt gene function and sometimes harm health. Most often that happens when reading frames are offset. And disorders that have been associated with these types of effects relate to behavioral disorders, especially autism and schizophrenia. Keep in mind that the degree that a mutation will alter a phenotype depends on a few things. One, where in the DNA sequence the actual change occurs, and how the mutation affects the way that the protein is either folded or formed, or its activity and abundance. So, for example, a mutation that replaces an amino acid with a very similar one in terms of charge or shape probably won't affect the phenotype that much because the overall shape of the protein may not be altered as severely. In addition, if the mutation is located or positioned in a region of the protein that isn't as critical to its function, so let's say it's not within the active site, but it's in a different location of the protein, it may not have as severe effects as an amino acid change right at that active site that's important for protein function. Globin gene mutations offer us some insight into the importance of position and the type of mutation in terms of effects on phenotype. So there are hundreds of globin gene mutations that are known to science, and these mutations have a wide range of associated phenotypes, as you can see in this table. So we've got the phenotypes, the name of the condition, and exactly what the mutation does in this table. So phenotypes can range from clinically silent to impacts on the ability of hemoglobin to bind oxygen, delivering it to the body. Anemia, so issues of transporting iron throughout the system, as well as protection against malaria, which I've already covered. So these instances may or may not involve that sickling shape of the blood cells as well. So your text has some really interesting kind of case studies describing these different mutations and associated conditions if you're interested in reading about them. At the end of the day, mutations are pretty rare. One of the reasons is that DNA polymerase is really good at detecting error and damage. So replication is pretty accurate. Only 1 in 100 million or so bases are incorrectly incorporated during the replication process. There are other types of DNA repair. We're going to focus on a couple of examples. So your book mentions one type that we as humans don't have. It's called photoreactivation repair. So these are enzymes that use light energy to actually break extra bonds that start happening in dimers. So dimers are really common when an organism is exposed to lots of UV damage. We're going to focus on two different examples here that we see in humans. The first one is called excision repair, and we have two types. We're also going to talk about mismatch repair. We humans have two different types of excision repair. We have nucleotide excision repair, which involves up to about 30 nucleotides, so larger chunks of errors. And the errors are caused by several different factors that can include exposure to chemical carcinogens or UV radiation or oxidative damage. So this involves a large number of different proteins that are actively involved in kind of working together cooperatively to detect and fix repairs. So in this example, we see how UV radiation causes these dimers, so kind of this bubble in the DNA sequence. Repair enzymes detect, remove incorrect sequences. DNA polymerase can come in and replace the correct nucleotides where they should be. Ligase, which we are well familiar with, comes in and conceals things back to original condition. We also have base excision repair. So this is kind of a smaller scale repair because it only involves up to 1 to 5 nucleotides at a time, but it's used to specifically correct damage that was caused to exposure to free radicals of oxygen. So these free radicals are forms of oxygen that arise during chemical reactions, and they damage DNA. So any of our genes that are actively transcribed are exposed to greater extents of oxidative damage from these free radicals. So this excision repair targets those types of damages. Finally, we also have a mechanism called mismatch repair. So enzymes detect nucleotides that are not properly paired in any newly replicated DNA. That incorrect base is first removed, and then it's replaced with the correct base. So this is an example where we have proofreading leading to the detection of these mismatches so that other enzymes can come in and snip and the correct base can be replaced. As we've learned with any of our other processes, DNA repair can have its own genetic disorders. So if a mutation occurs in any of the genes that encode these specific DNA repair proteins, now we've covered many different types that are important to these cascading cooperative mechanisms that can cause problems with DNA repair. Your text covers several different examples, and just one that I'm covering here is protein P53. It's a protein that monitors DNA repair, and if the damage is too severe to be repaired within a DNA sequence, this protein triggers programmed cell death. So if there is a mutation that affects the proper functioning of P53, what happens is that these severe mutations aren't lost. So they're maintained in the body and they can actually accumulate to severe levels.